everyone. Today we're diving into a crash course on control of gene expression.
It sounds a bit technical right out of the gate, I know, but if you stick with us, you'll see that this is actually one of the most fundamental concepts in all of biology.
Right.
We're essentially talking about the operating system of life.
The operating system, that is a big claim. But before we get into the nuts and bolts of it, I want to start with the core mystery.
Yep.
It's something I think we all just kind of take for granted. If you look at a kidney cell from your body under a microscope and then you look at a bone cell, they look completely different.
Oh, absolutely. I mean, functionally, morphologically, they are worlds apart. One's filtering blood, the other is building a rigid matrix.
But, and here's the kicker, they have the exact same instruction manual.
Mm-hmm.
Every single cell in your body contains the exact same DNA. So the question is, how does a kidney cell know to be a kidney cell and not a bone cell if they're reading the exact same book?
That is the core question. And the answer lies in what we call gene expression. It turns out it's not about what genes you have, since all your cells have all your genes.
Right. They all have the whole library.
It's about which genes are turned on and which ones are turned off.
So it's like a kidney cell has just checked out the How to Be a Kidney books and is completely ignoring the How to Be a Bone section.
That's a perfect way to visualize it. And today, we're going to trace exactly how that works. We'll start simple with bacteria,
Okay.
then move to our own complex chromosomes, follow the whole regulatory pipeline, and finally see how it all builds an organism.
All right, let's do it. We're starting with bacteria, specifically our old friend E. coli.
Yes, Escherichia coli. It's likely living in your intestine right now.
A glamorous life.
It's a chaotic one. I mean,
Yeah.
think about it. The environment for that bacteria changes constantly based on what you eat.
Right. So if I drink a glass of milk.
Suddenly, there's lactose everywhere.
Yeah.
But if you eat a salad, the whole resource landscape changes. The bacteria has to adapt.
It can't waste energy.
Precisely. Efficiency is everything. If there's milk, it needs enzymes to digest lactose. But if there's no milk, making those enzymes is a huge waste of energy.
So how does this tiny organism know when to turn the milk digestion genes on or off?
It uses something called an operon. An operon is basically a cluster of related genes, like the 3 needed for lactose, all controlled by a single on-off switch.
One switch for the whole factory line. That's smart.
Very. And the switch itself is a piece of DNA called the operator. It sits right between the promoter, where the machine
The
reattaches,
starting line.
and the genes themselves. So it's like a gate on a train track.
Okay, I can see that.
Now, if there's no lactose around, the default state is off. A protein called a repressor binds right to that operator.
It closes the gate.
It physically blocks the track. The RNA polymerase can't move. No transcription.
But then I drink a milkshake. Lactose floods the system.
The lactose molecules themselves are the key. They bind to that repressor protein, and when they do, they change its shape.
And it lets go.
It can't hold on anymore. It just falls off the DNA.
The gate swings open.
And the polymerase train goes right down the track, transcribes the genes, and the bacteria starts digesting your milkshake.
Wow. The food itself unlocks the genes to digest it.
It's elegant, isn't it?
Yep.
And that's an inducible operon, normally off, but you can turn it on.
And there's an opposite version.
There is a repressible one, like the trepoperon, which makes tryptophan, an amino acid.
Something the bacteria needs all the time?
Usually, yeah. So this factory is normally on. But if you, say, eat a big turkey dinner, suddenly there's tryptophan everywhere.
No need to make it if it's free.
Exactly. So the tryptophan molecule itself binds to the repressor and activates it.
Ah, so it helps the repressor clamp down and shut the factory off.
You got it. It's the opposite logic. Same goal. Efficiency.
Now, is it always about blocking things? Or are there proteins that, help things along?
Oh, yeah. Those are called activators. They bind to the DNA and help the RNA polymerase get started. Sort of like a volume boost.
Okay, so that's bacteria. Neat little switches. But we are a lot more complicated.
Just a bit. And that brings us to the next big challenge. Storage. If you stretched out the DNA in just one of your cells, it would be about 2 meters long.
2 meters in a microscopic nucleus.
Yeah. It's an incredible packing challenge. To do it, our cells wrap the DNA around tiny proteins called histones.
Like thread around a spool?
Very much so. These spools are called nucleosomes, and they look like beads on a string. Then those beads coil and loop and fold again and again into a chromosome.
Okay, so that's storage. How does that connect to control?
Well, think about that train track again. What if the track is wound up in a super tight ball of yarn?
The train can't run on it. It can't even get to it.
Exactly. DNA packing is a form of gene control. If a gene is in a tightly packed region, it's effectively turned off.
And the cell can control how tight it is.
It can. By adding little chemical tags, adding a methyl group, for instance, usually tightens the packing, turning genes off. Adding an acetyl group tends to loosen it up.
It's like a chemical open or close sign.
Precisely. Which brings us to a really cool, visible example of this.
The tortoiseshell cat. I love this.
It's the best example of something called X inactivation. So female mammals have 2 X chromosomes, XX. Males only have 1.
Right.
If females use both, they'd get a double dose of all those proteins, which could be a problem. So very early in development, 1 X chromosome in every single cell gets randomly shut down.
Shut down how?
It gets compacted into a super dense, inactive knot. We call it a Barr body.
And it's random, like a coin flip in each cell.
Completely random. Now, in cats, a gene for fur color is on the X chromosome. So one X might have the allele for orange fur, and the other might have the allele for black fur.
So a female cat could have both versions.
And if she does, she becomes a mosaic. In some patches of her skin cells, the orange X was left on, so you get orange fur. Right next to it in another patch, maybe the black X was left on.
So the Cas coat is literally a map of which X chromosome got turned off in which cells.
That's exactly what it is. There's a visual map of gene regulation.
That is wild. And this is related to a term I hear all the time now, epigenetics.
Yes, epi means above or on top of. Epigenetics is the inheritance of traits that are transmitted by mechanisms not directly in the DNA sequence.
Wait, inheritance that's not in the A's, C's, T's, and G's.
Right, those methylation marks we talked about, the off switches. When a cell divides, it can pass those marks to its daughter cells. The DNA sequence is identical, but the instruction to keep a gene off is inherited.
That's how a skin cell knows to make more skin cells.
Yeah, it remembers its epigenetic instructions. It passes down the bookmarks, not just the books.
Okay, so we've unpacked the DNA. Now we actually have to start transcription. Is it as simple as the bacterial gate opening?
Not even close. The first RNA copy has a lot of, well, junk in it, non-coding sections called introns. The cell has to cut those out and stitch the good parts, the exons, together.
But, and this blew my mind, you can stitch them together in different ways.
Alternative RNA splicing. This is a huge deal. It means a single gene can produce many different proteins, just depending on which exons you include in the final message.
Which is how humans, with about 21,000 genes, can be so much more complex than a worm with the same number.
Exactly. We get something like 100,000 different proteins from those 21,000 genes. It's incredibly efficient.
It's like Legos. The same bricks can build a car or a plane.
Perfect analogy. And then there are other valves. How long does the mRNA message last? In bacteria, it's minutes. The mRNA for hemoglobin in our red blood cells lasts for weeks.
More protein from the same message.
You got it. And even after the protein is built, it might not work yet. Think of insulin. It's made as one long, inactive chain. You have to cut a piece out of the middle to activate it.
So many layers of control.
It's all about getting the right amount of the right protein at exactly the right time.
Which brings us to that so-called junk DNA.
Ah, yes. The myth. For years, we thought 98% of our DNA was useless. We were very, very wrong.
So what is it doing?
A huge chunk of it is transcribed into non-coding RNAs. The most famous are microRNAs, or miRNAs.
And they do what, exactly?
They're like gene expression police. They are tiny RNA molecules that hunt down specific messenger RNAs. If they find a perfect match, they signal for that mRNA to be destroyed.
They silence it.
They silence it. If it's a partial match, they just block it from being translated. This process, RNA interference, was a Nobel Prize-winning discovery.
And now scientists can use this, right, to turn genes off on purpose.
And we can. We can design a synthetic small RNA to shut off any gene we want to study. It's like pulling a fuse in a house to see which lights go out. Very powerful tool.
So this system of control, it's what builds a body from a single cell.
It is. Which brings us to the fruit flies.
Yes. The fly with legs growing out of its head.
A mutant called Antennapedia. It's a shocking image, but it taught us about a critical class of genes.
Oh, meiotic genes.
The master control genes. These genes don't just make one protein. Their job is to tell a whole segment of the body what to become. They say this segment is a head, or this segment is a thorax.
So a single mutation in one of those genes can just swap a whole body part.
It can, because it triggers a whole cascade of other genes. The homeotic gene is like a general giving an order to an entire division of an army.
A developmental domino effect.
A very precise one.
So we've talked about how all this works, but how do scientists actually see it? How do you know which of those 21,000 genes are on or off in a cancer cell, for example?
For that, you use a tool called a DNA microarray, or a gene chip.
Walk us through it. It looks like a grid of glowing dots.
That's what it is. Imagine a glass slide with thousands of tiny spots. In each spot, we've fixed a bit of DNA from one specific gene.
So you have the whole genome laid out on a grid?
Essentially, yes. Then you take your samples, say from a tumor and from healthy tissue. You isolate all the messenger RNA.
The messages from the genes that are currently on?
Exactly. Then you make glowing DNA copies of those messages. You label the cancer ones red and the normal ones green.
Okay, red for cancer, green for normal.
You wash this mix of glowing DNA over the chip. If a gene was active in the cancer cells, its red-tagged copy will stick to its spot on the grid.
And the spot will glow red.
And if it was active in the normal cells, it will glow green.
And what if it's yellow?
Yellow means both red and green are there. The gene was active in both types of cells.
So you get an instant snapshot of the entire gene expression profile of a cell.
It's revolutionary for medicine. We can classify cancers not just by how they look, but by which genes are driving them. It leads to personalized treatment.
And this all comes back to cells talking to each other, right? That's the final piece.
Cell signaling, the signal transduction pathway. A molecule docks on the outside of the cell, triggers a relay inside, and that signal travels to the nucleus to flip a transcription switch.
It's the trigger for the whole process.
It's what connects the outside world to the genes inside.
So to bring this all together, we start by asking why a kidney cell is different from a bone cell.
And the answer is this incredible multi-layered system of control. From the simple switches in bacteria to the complex folding, splicing, and signaling in our own bodies.
It's not just about having the genes, it's about how you play them.
That's a beautiful way to put it. And I'll leave you with this final thought. We now have tools to read these switches with microarrays and to flip them with RNA interference. We map the genetic code. But what if we could truly master the epigenetic code?
What do you mean?
If we could learn to safely and precisely flip these switches ourselves, could we turn off the genes that drive cancer? Could we turn on repair genes to fight aging?
Could you tell a cell to become young again?
That is the frontier, and it's closer than you might think.
Wow, a lot to think about. That's our deep dive on gene expression. Thanks for listening.